Hello everyone, welcome to MedLocus. In this video, we will explore the fascinating mechanism of hydrochloric acid secretion, a vital process in our digestive system. In this session, we are going to witness the dance of hydrogen ions and chloride ions as they travel across number of channels and pumps, converging into the canaliculus of special cell of our stomach, that is parietal cell, which is the epicenter of acid production. So, without further ado, let's get started. Before moving to know about the mechanism of HCl secretion, let us first try to see our stomach where the acid is produced. This is the picture of stomach representing the cut section of its wall. The cut section shows numerous gastric glands in its mucosa with gastric pits. From the gastric pits, the secretion of gastric glands, including the strong acid, is secreted into the lumen of stomach. Now, from this, let us extract a single gastric gland for better visualization. Can you see the highlighted parietal cell in this picture? Don't forget, parietal cells are also called axanthic cells. Within a single gastric gland, you can see there are several parietal cells, right? Out of them, let us again extract a single parietal cell now. Can you see? This cell has two surfaces. One is basolateral surface and next one is luminal or apical surface. Basolateral surface faces extracellular side and is in contact with the surrounding tissues and blood vessels. Luminal or apical surface faces the lumen of stomach. Basolateral surface has pumps like sodium-potassium ATPase pump, chloride bicarbonate exchangers, and apical surface has conductance channels and very important hydrogen-potassium ATPase pump, which we will talk in later portion of this video. Let us now zoom in this cell. Here we can see water, that is H2O in cytoplasm of parietal cell of gastric gland. This water or H2O dissociates in the cytoplasm of parietal cell, generating hydrogen ions and hydroxyl ions, that is H plus ions and OH minus ions. After this, CO2, that is carbon dioxide, enters parietal cell by diffusion, and it is the slowest process of entire gastric acid production. The production of gastric acid is dependent on the diffusion of carbon dioxide through the basal surface of parietal cell, which serves as the rate limiting step in acid synthesis. Carbonic anhydrase enzyme catalyzes the rapid combination of OH minus with CO2 to form carbonic acid, that is H2CO3. Again, the carbonic acid dissociates into bicarbonate ions, that is HCO3 minus, and hydrogen ions, that is H plus ions. Now, HCO3 minus ions, that are bicarbonate ions, are transported out of the parietal cell into the blood in exchange for chloride ions. This increases intracellular level of chloride ions. Excess bicarbonate ions in plasma results in slight elevation of blood pH, which is known as alkaline tide. This is just a temporary process, and pH is again restored to normal pH by maintenance of acid-base balance. Intracellular fluid or cytoplasm of parietal cell is rich in sodium ions and ATP due to numerous mitochondria in parietal cell. 3 sodium ions are transported out of the parietal cell catalyzed by sodium-potassium ATPase pump present in basolateral membrane in exchange of 2 potassium ions from the extracellular fluid or plasma of gastric vessels. This occurs repeatedly and ultimately leads to increased intracellular level of potassium and chloride ions. Now, these ions are transported into the lumen of canaliculus through conductance channels. Chloride ions are passed through chloride channels and potassium ions are passed via potassium channels. This leads to increased level of potassium and chloride ions in canaliculus of parietal cells. Now, increased amount of potassium is pumped back to the cell from canaliculus by proton pump or H plus K plus ATPase pump of apical membrane. But Before we talk about mechanism of these proton pumps, let us first discuss how do they come to the apical membrane. For that, we need to know some receptors present in parietal cells like M3 receptors, S2 receptors, CCKV receptors, which are activated by acetylcholine, histamine and gastrin respectively. After activation of these receptors, there is fusion of tubular vesicles containing hydrogen-potassium ATPase pump into apical membrane. Now, let us take a single proton pump and learn its mechanism. The proton pump or H plus K plus ATPase located in the canalicular membrane actively transports hydrogen ions out of the parietal cell into the lumen in exchange for potassium ions. One ATP is utilized to transport one hydrogen ion into luminal site and one potassium ion back to the cell. Finally, after several times of transport process, there is increased concentration of hydrogen and chloride ions in canaliculus. Also, accumulation of osmotically active hydrogen ions generates an osmotic gradient, causing outward diffusion of water, forming strong solution of hydrochloric acid in canaliculus. Thus, final secretion from the canaliculus contains water, hydrochloric acid at a concentration of about 150 to 160 milliequivalent per liter, potassium chloride at a concentration of 15 milliequivalent per liter, and a small amount of sodium chloride as well. Finally, let us conclude this video by listing some important functions of HCl and stomach.